I'd like to introduce our final speaker of the session, Dr. Tyler Jacks, who's coming from MIT, and he'll be talking about modeling the RAS pathway in the mouse and developing therapies using these animals.
Thanks, Karen. Pleasure to be here. Thanks, Bruce, for the invitation. That was a very provocative talk, Pier Paolo, but very unsettling. I found myself getting queasy just thinking about it, actually. So I am going to talk about RAS-driven models today, but actually more broadly than just with respect to therapeutics. I'm going to touch on certain new projects related to cancer biology, which also have implications for therapy. My lab is motivated to study cancer development in situ. Although we know that cancers arise through alterations in cellular genes like oncogenes and tumor suppressor genes, we also know that these cells interact with cells of the host in important ways that both promote and inhibit tumor progression. These interactions influence the biology of the tumor cell, its evolution, and ultimately its response to therapy. In addition, tumor cells move from their primary site in the process of metastasis. This is still a poorly understood process, but clearly very important clinically, with 90% of cancer deaths being attributed to metastasis. We think that ultimately we'll need to study this process in vivo as well to understand it in detail. So with those principles in mind for many years, like with Pier Paolo and with Len, we've chosen to look in vivo. And for us, the model system of choice is the mouse, largely because we can manipulate the genes of mice with ease and make mutations of cancer genes of interest in greater and greater detail, and to mutate those genes with more precision with respect to the cell types and the timing in which they occur. Now, importantly, the mouse has received some criticism over the years, including in this famous article by Clifton Leaf in Fortune magazine several years ago, in which he objected in many ways to the approaches that we've been taking over the years in our work in cancer research, the fact that we were losing the war on cancer, and a main villain in that story was actually the laboratory mouse. This is a quote from that article. It says, if you look at the millions of mice that have been cured of cancer and compare it to humans, you realize there just has to be something wrong with those models. This quote was actually unattributed. I actually think it was Bruce Chabner who made the quote, but in fact, it's a fair statement. It's a fair statement, but it doesn't really refer to the genetically engineered models that I work on, that Pier Paolo works on, that many others in the community do, but rather to the immunocompromised mouse, in which one grows human cancer cells subcutaneously in a very artificial environment, which doesn't in any way recapitulate those important interactions that I introduced to you on the previous slide. So we hope, and I underscore the word hope, that the new age models with genetically engineered mutations and cancer-relevant genes in the right cell types and so forth and so on will do better. This is an experiment that's ongoing in many of our laboratories, and Pier Paolo is leading one such effort in the mouse hospital experiment that he described to you briefly. I think, and the early indications are, that these models will be better, will be more predictive, will be more useful, but we'll see. For us, we use genetic engineering techniques, and I won't talk about them in detail, but I'll mention a few details in a moment, to create mutations in genes of interest, as I said, and watch the process unfold in mice, and we can then access tissue and look at details of tumor progression, and I'll give you a story about that in a moment. Once these animals have been built, we hope that they will be useful for practical purposes like treatment, but also prevention and early detection. Obviously, we need to be able to detect cancers at earlier and earlier stages, and we can test drive some of these methods and maybe discover new techniques utilizing these mouse model systems.